Here is what we know about time. It moves in one direction. Every cell in every organism that has ever lived on this planet, every bacterium, every whale, every human being, is built around that single, inescapable axiom. You are born, you grow, you age, you die. The molecular clock inside each of your cells ticks forward, and it does not tick back. This is not a philosophical statement. It is a mechanical fact encoded into the architecture of your DNA. Your telomeres, the protective caps at the end of every chromosome, shorten with every cell division, like a fuse burning, and when they run out, the cell stops dividing. This is called senescence, and senescence leads to one place. Every organism on Earth obeys this rule. Every organism except one. Its name is Turritopsis dohrnii. Scientists call it the immortal jellyfish. But that name is almost embarrassingly small for what this organism actually is. This is not a creature that lives a long time. A Greenland shark lives 500 years. A bowhead whale, 200. Longevity is easy. It is simply a matter of running the metabolic engine slowly enough. What Turritopsis dohrnii does is something categorically different. When this animal is damaged, starving, or simply old, when any other organism would begin the process of dying, Turritopsis dohrnii does not die. It goes backward. It reverts its own body, cell by cell, to the juvenile state it existed in at the beginning of its life. And then it starts again. Not a copy, not offspring. The same organism, biologically young, fully reset. This is not a metaphor. This is not science fiction. And this is a mechanism that has been observed in the laboratory dozens of times, sequenced at the genomic level, and published in the Proceedings of the National Academy of Sciences. And we still do not fully understand how it works. The ocean has been here for 3.8 billion years. Life has existed in it for almost all of that time. Creatures have evolved eyes, jaws, camouflage, venom, electroreception, echolocation, every imaginable weapon for the business of surviving. But only one organism found a solution to the oldest problem of all, and it is the size of a lentil. To understand what Turritopsis dohrnii does, you need to understand what your cells cannot do. Every cell in your body contains the same complete genome, the full instruction manual for building an entire human, but each cell reads only the chapter relevant to its job. A liver cell reads the liver chapter. A neuron reads the neuron chapter. The other chapters are locked, silenced by a system of molecular switches called epigenetic markers. This locking is permanent. Or it should be. In 2006, the Nobel Prize in Physiology was awarded to Shinya Yamanaka for discovering that 4 specific transcription factors, proteins that control gene expression, could force an adult specialized cell to abandon its identity and revert to a pluripotent state. A cell capable of becoming anything. The scientific world called this discovery revolutionary. They called those factors the Yamanaka factors. In humans, achieving this in a living organism without triggering cancer remains one of the central unsolved problems of regenerative medicine. Turritopsis dohrnii does it automatically, under stress, in the open ocean, and the process is called transdifferentiation. A fully specialized adult cell transforms directly into a different cell type, without first becoming a stem cell. A muscle cell becomes a nerve cell. A nerve cell becomes an epithelial cell. The organism does not merely heal, it restructures itself from the inside out, erasing its developmental history and rewriting it from the beginning. A 2022 landmark study published in PNAS, led by researchers at the University of Oviedo, sequenced the complete genomes of both Turritopsis dohrnii and its mortal close relative, Turritopsis rubra. What they found was architectural. The immortal jellyfish carries duplicated copies of genes involved in DNA repair, telomere maintenance, and oxidative stress response. Where T. rubra has one copy, T. doherni has two. The evolution's equivalent of engineering redundancy. Backup systems for backup systems. More critically, the Oviedo team identified a specific mutation in a gene called POT1, protection of telomeres 1. In humans, POT1 acts as a brake on telomerase, the enzyme that rebuilds telomere length. When POT1 is impaired in humans, telomeres can elongate uncontrollably, a precursor to cancer. But in T. doherni, the mutation appears to have been co-evolved with compensating molecular changes, creating a system that maintains telomere length without triggering uncontrolled cell division, the biological equivalent of a brake that also functions as an accelerator, depending on context, depending on what the organism needs. But the most astonishing component of this machinery is not the final result. It is the intermediate step. When Turritopsis dohrnii undergoes life cycle reversal, it does not transform directly from adult medusa to juvenile polyp. It passes through a transitional state called the cyst, a temporary structure that has no analog in any other organism on Earth. The cyst is, biologically speaking, an almost featureless cluster of cells. Under the microscope, it is indistinguishable chaos, but inside that chaos, the most sophisticated cellular reprogramming process in the animal kingdom is running at full capacity. A 2021 transcriptome analysis published in Genome Biology and Evolution and a July 2025 preprint on bioRxiv expanding those findings found that during the cyst stage, the jellyfish activates gene networks associated with pluripotency, DNA repair, telomere maintenance, and longevity. Sirtuins, proteins that regulate cellular aging and are the target of enormous pharmaceutical investment in human longevity research, are specifically upregulated. Heat shock proteins, which protect the cellular machinery from damage, surge. The tumor suppressor BRCA1, which repairs DNA and prevents erratic cell division, is strongly expressed. The cyst is not dying, it is performing a comprehensive systems overhaul, and it completes the entire process in approximately 24 hours. Consider the human comparison. Our best current approach to cellular reprogramming, partial Yamanaka factor induction, takes weeks in laboratory conditions, requires precise chemical control, and carries significant risk of oncogenic transformation. One miscalibration and a cell becomes a tumor. And Turritopsis dohrnii triggers a more complete version of this process in a living organism under environmental stress, in open salt water with no laboratory intervention. Evolution spent 600 million years engineering this, and whatever it built, it works. The body of Turritopsis dohrnii is approximately 95% water. There is no skeleton, no brain, no centralized nervous system in any sense that would feel familiar to a vertebrate biologist. What it has instead is a nerve net, a diffuse web of neurons distributed across the entire body surface. There is no command center, no hierarchy. Sensation and response are handled in a distributed fashion, closer to the architecture of an internet than the architecture of a mammalian nervous system. This might seem like a limitation. In evolutionary terms, it is a design choice. The tentacles of T. dohrnii are lined with nematocysts, among the fastest mechanical actuators in the animal kingdom. Each nematocyst is a microscopic pressurized capsule containing a coiled barbed filament bathed in paralytic venom. When a prey organism contacts the trigger, the internal pressure, equivalent to 150 atmospheres, fires the filament at speeds exceeding 60 kilometers per hour. The entire process occurs in 700 nanoseconds. For context, a bullet travels from a handgun barrel in roughly 1 millisecond. The nematocyst fires 1,400 times faster. This is not aggression. It is precision engineering operating at the boundary of what chemistry allows. And it is the same basic design that has persisted, largely unchanged, for over 500 million years. The distributed nerve net processes this without a brain. Not despite the absence of centralized intelligence, but because of it. Distributed processing means no single point of failure. Damage one region of the nerve net, and the rest continues functioning. In evolutionary engineering terms, this is called fault tolerance. The human nervous system, for all its complexity, is catastrophically fragile by comparison. A stroke disabling a few cubic centimeters of prefrontal cortex can eliminate language, personality, and motor function simultaneously. Turritopsis dohrnii does not have this problem. Nothing in biology is free. Every adaptation carries a cost, and evolution is a ruthless accountant. The transdifferentiation mechanism of Turritopsis dohrnii is metabolically expensive, and the cyst stage consumes significant energy precisely because it is doing the most complex thing any cell can do: abandoning one identity and constructing another. DNA synthesis pathways are upregulated. Protein refolding machinery runs at capacity. Telomere maintenance enzymes are active. But evolution made a calculation, and the calculation went like this. The cost of maintaining a cellular reset mechanism is lower than the cost of dying. In the Mediterranean, where T. dohrnii originated, conditions fluctuate violently. Water temperature swings, salinity shifts, food availability collapses unpredictably. A small organism with no skeleton, no shell, no speed, and no size has very few options when the environment turns hostile. Death is one option. Dormancy is another. But reversal, returning to a sessile polyp on the seafloor and waiting, is a third, and it is the most radical. The life cycle of T. dohrnii follows the standard hydrozoan pattern. Fertilized egg becomes planula larva, planula anchors, and becomes a polyp colony buds, and releases free-swimming medusae. In all other jellyfish, the medusa stage ends in reproduction and death. The polyp colony they grew from continues, but the medusa, the free-swimming individual, does not. In T. dohrnii, the medusa can return to the polyp, not metaphorically. The organism physically undoes its own adult development. This has been confirmed in laboratory settings, where individual medusa have been observed reversing their life cycle multiple times. In one documented case at Shin Kubota's laboratory in Japan, the same colony reversed repeatedly over 4 years. This is the evolutionary gamble. Invest heavily in the machinery of reversal. Pay the energy cost every time conditions force a reset. But never die of old age. The trade-off is constraint, not freedom. Turritopsis dohrnii cannot grow large, cannot develop sophisticated predation strategies, cannot build anything approaching a complex nervous system. It bought one thing, and it bought it absolutely: the ability to treat death as a temporary condition. Turritopsis dohrnii began as a Mediterranean species. It is now everywhere. The mechanism of dispersal was not natural ocean current. It was ballast water, the seawater that cargo ships take on in one port and discharge in another, used to maintain stability. Tucked into those billions of liters of water, invisible, indistinguishable from the ambient marine environment, were polyp colonies of T. dohrnii. They were discharged on every inhabited coastline on the planet. Because the organism can survive almost any stress condition by reverting to its polyp form, it was extraordinarily well suited to this accidental global dispersal. A temporary cold, a temporary food shortage, a temporary period of darkness in a ballast tank, any of these conditions that would kill most marine organisms could simply be waited out. Turritopsis doharnii has now been documented in the Mediterranean, the Caribbean, the Pacific coast of Japan, Panama, Australia, and along the eastern coast of North America. It is one of the most widely distributed marine organisms on Earth, and in most of those locations, it is an invasive species, not a predator, not a competitor for large prey, but a presence occupying the plankton layer, quietly running its metabolic cycles, and occasionally, when conditions demand it, disappearing and reappearing months later, as if nothing happened. An organism that cannot be killed by old age, that can compress itself into a dormant state to survive almost any environmental stress, and that has now colonized every major ocean on the planet. Evolution did not plan this, but it built the tools that made it possible. Here we need to be precise, because the word immortal invites a misunderstanding. Turritopsis doharnii cannot be killed by aging. Its biological clock can be reset, but it can be killed by everything else. Fish eat it. Sea turtles eat it. Other jellyfish eat it. The ocean is not short of organisms willing to consume a 4-millimeter translucent bell with no defensive armor. Most individuals never complete a single life cycle reversal. They are consumed before the crisis that would trigger it. The mechanism exists, and it functions, but in the open ocean, a creature must survive long enough and be robust enough through the actual reversal process to benefit from it. This is the paradox at the heart of T. doharnii's existence. It possesses the most sophisticated anti-aging mechanism ever discovered in an animal, and it dies constantly. Not from age, but from the ordinary brutality of the food chain. Biological immortality is not the same as invulnerability. Turritopsis doharnii solved one problem, the most profound biological problem there is, and remained entirely exposed to all the others. And in this sense, it is the perfect metaphor for what evolution actually does. It does not optimize globally, it patches locally. It solves the problem that is killing you right now and ignores the one that will kill you next. The discovery of Turritopsis doharnii's life cycle reversal was an accident. In the late 1980s, two marine biologists, Giorgio Bavistrello and Ferdinando Sommer, left a jar of adult T. doharnii medusae in a laboratory over a weekend without feeding them. When they returned, the jar contained not dead medusae, but a polyp colony. The medusae had not died. They had reverted. The paper documenting this was published in 1996. For most of the following decade, it was noticed only by specialists in hydrozoan biology. The rest of biology continued under the assumption that aging was, at the cellular level, irreversible. This assumption was not unreasonable. It was based on 150 years of accumulated evidence from every model organism in the field: yeast, nematode worms, fruit flies, mice. All of them age in one direction. All of them confirmed the same underlying logic. What T. doharnii revealed was that this logic was not universal. It was simply the solution that most animals happen to settle on. The problem of how to manage cellular resources over time had, apparently, more than one answer. The reason it took so long to recognize the significance of this is instructive. Scientists study the organisms they have the tools to study. Turritopsis doharnii is tiny, fragile, difficult to culture in the laboratory, and found in an environment, the open plankton layer, that is extraordinarily difficult to observe systematically. It did not appear in any of the canonical model organism lists that 20th century biology organized itself around. We almost missed the most important biological discovery of the century because the animal was 4 millimeters wide, and nobody was looking at it carefully. In 2022, the Oviedo study gave us something we had never had before: the complete genomic blueprint. The findings were not ambiguous. Compared to its mortal relative Turritopsis rubra, T. doherneii had twice the number of genes associated with DNA repair, extra copies of thioredoxin and glutathione reductase, proteins that neutralize the oxidative damage that accumulates in aging cells, expanded copies of GLI3, a transcription factor involved in stem cell maintenance, and that critical POT1 variant, are allowing telomere maintenance without triggering the uncontrolled growth that kills human cells when similar mutations occur. But perhaps the most remarkable finding was what happened to polycomb repressive complex 2 during life cycle reversal. PRC2 is one of the primary mechanisms by which cells enforce their differentiated identity, the molecular lock that keeps a liver cell reading only the liver chapter. During T. doherneii reversal, PRC2 targets are silenced. The locks come off. Simultaneously, pluripotency targets, genes that define undifferentiated, potentially totipotent states, are activated. The jellyfish is, in the precise technical language of cell biology, performing the equivalent of the Yamanaka reprogramming protocol. Spontaneously. Repeatedly. In a living animal. A July 2025 preprint from bioRxiv extended these findings further, profiling the expression of sirtuin genes, telomere maintenance factors, and heat shock proteins across all life cycle stages using a supertranscriptome approach. The data confirmed that T. doherneii reversal engages a coherent, integrated genetic program, not a random stress response, but a coordinated sequence of molecular events that evolution has refined over hundreds of millions of years. This matters for human medicine in ways that are genuine, but must be stated carefully. We will not inject Turritopsis doherneii genes into humans and achieve immortality. The gap between a 600-neuron, radially symmetric marine invertebrate and a mammal with 86 billion neurons is not a gap that genomic borrowing will bridge, but the specific molecular mechanisms T. doherneii uses, the particular configuration of telomere maintenance, the specific silencing of PRC2, the precise cascade of pluripotency gene activation, these are legitimate research targets. Understanding how an animal performs spontaneous, safe, complete cellular reprogramming without triggering cancer could inform how we approach cellular senescence, regenerative medicine, and the treatment of age-related diseases in ways that are not currently available. The jellyfish did not evolve these mechanisms for our benefit, but they are there, and they work, and we are only beginning to read them. There is a version of this story in which Turritopsis doherneii is a curiosity, a strange edge case in the catalog of marine biology, an organism that found a loophole. But I think that reading misses something important. And we have built our entire understanding of aging, our philosophy of what it means to be a living thing in time, on the assumption that the biological clock runs in one direction. That assumption is woven into our medicine, our ethics, our art. When we talk about fighting aging, or accepting aging, or embracing the finite, we do so on the premise that these are the only options available, that this is what biology permits. Turritopsis doherneii does not accept that premise. It never did. It found a third option 600 million years ago and has been exercising it quietly, in the open ocean, in complete indifference to our categories. We did not notice for most of our scientific history because we were not looking for it. We were studying animals that behaved the way we expected animals to behave, the ones that fit our models, and the ones we could relate to. A 4-millimeter transparent organism drifting through Mediterranean plankton did not fit any of those criteria. And so we almost missed the only evidence we have ever found that the fundamental rule of biological time is not, in fact, absolute. Somewhere in the Mediterranean, or in the waters off Japan, or in the Caribbean, a Turritopsis dohrnii is dying right now. Its cells are collapsing. Its body is folding inward. Everything it was is dissolving into a structureless cluster of matter, and in 24 hours, it will not be dead. It will be young. It will begin again, without memory, without history, without any apparent awareness of what it has just done. And that fact, more than any genome sequence, more than any laboratory observation, is the thing that should give us pause. And not because we can harness it, not because it will save us, but because it means the universe we thought we understood, the one with a fixed arrow of time and a final destination for every living thing, is slightly more strange than we believed. It always is.